Hello everyone, I'm Nicole Comfort, and today I will introduce you to our original article entitled Isolation and Characterization of Extracellular Vesicles in Saliva of Children with Asthma. Asthma is the most common non-communicable childhood disease, affecting approximately 8% of children worldwide and 12 to 15% of urban children in the United States. Asthma is a multifactorial disorder characterized by inflammation, airway remodeling, and airway hyperresponsiveness. While the clinical presentations may appear similar, evidence suggests that asthma may not be a single disease, but rather encompasses a group of heterogeneous endotypes with various etiologies and prognoses. Identifying biomarkers that can profile clinical subtypes early in the course of asthma is critical in applying tailored therapy, yet the utility of current biomarkers is very limited because they are either invasive or nonspecific and therefore unable to faithfully identify clinically meaningful subtypes. Extracellular vesicles, which I'll hereafter refer to as EVs, are attractive candidates for biomarkers of asthma endotypes. EVs are small, membrane-bound vesicles secreted by nearly all cell types. They have classically been referred to as exosomes or microvesicles, depending on their mechanism of release and size. While it was initially thought that EVs were simply a means of eliminating waste from the cell, we now know that they can also participate in intercellular communication via transfer of molecular material such as RNA, lipids, and proteins, and that they are important regulators of normal physiology as well as pathological processes. EVs and their cargo have been implicated in asthma. Also, EVs have been found in nearly all biofluids tested, including saliva. Regarded as the mirror of the body, saliva is an easily accessible biofluid that harbors constituents that provide sources for monitoring of health and disease states, including asthma. Yet, to the best of our knowledge, no study has investigated the characteristics of saliva EVs among patients with asthma. The aim of the present study is to describe the EVs isolated from cell-free saliva of children with asthma. Whole saliva was collected from 180 participants of the school inner-city asthma study. These samples were centrifuged to obtain cell-free saliva. Cell-free saliva EVs were isolated using ExoQuick TC from System Biosciences, and a pooled sample of the cell-free saliva EVs were examined in downstream analyses. The EVs were characterized using multiple orthogonal techniques in accordance with the International Society for Extracellular Vesicles minimal experimental requirements for the definition of EVs. Transmission electron microscopy was used to confirm the presence of EVs. Nanoparticle tracking analysis was used to estimate the EV particle size distribution. Single particle interferometric reflectance imaging sensing, or SP-IRIS, and immunocapture with fluorescence run on the ExoView R100 platform was used for sizing, counting, and tetraspan and profiling of EVs. Capillary immunoassays run on the WES were used for protein quantitation of the tetraspanins CD9, CD63, CD81, the cell adhesion molecule ICAM1 and endosome protein annexin A5, and intracellular proteins underrepresented in EVs as negative controls. Transmission electron microscopy confirmed the presence of EVs of diverse sizes. Capillary immunoassays confirmed the presence of EV-associated proteins and indicated limited cellular contamination, denoted by the absence of the endoplasmic reticulum protein calnexin and Golgi-associated protein GM130. As others have also reported, there were discrepancies in the EV sizing and enumeration across platforms. For instance, fluorescent nanoparticle tracking analysis detected particles with a mode diameter of about 90 nanometers, whereas SP-IRIS reported sizes of about 55 to 60 nanometers that more closely approximated the transmission electron microscopy results. Consistent with protein immunoassay results, immunocapture with fluorescence on the ExoView R100 platform showed that the majority of these EVs were CD9 and CD63 positive, with little expression of CD81. EVs from cell-free saliva can be isolated using a high-throughput method that can be scaled to large epidemiological studies. To our knowledge, we are the first to characterize cell-free saliva EVs from patients with asthma. The use of cell-free saliva EVs as potential novel biomarkers in asthma warrants further attention and opens a new avenue of research for future studies. This is just a brief highlight of the content presented in our article. I hope you will enjoy reading more details, and I would like to thank you all for your attention. Please contact myself, or the Laboratory of Precision Environmental Health at Columbia for questions.